Hi friends, welcome all of you to the class of gingival epithelium. The last class was all about the anatomy of gingiva. For those who have not seen that, you go and check it out and then you come to this class. This is the diagrammatic picture which I showed in the last class. So you can see, this is the part of gingiva where this is epithelium and this is connective tissue. Epithelium is the covering of the gingiva. Inside we can see the connective tissue, and this is the outer epithelium. This epithelium covering the outer part is the outer epithelium, and last class we discussed that this is a sulcus. So the epithelium covering the sulcus is known as the sulcular epithelium, and this is the junctional epithelium which attaches to the tooth. So the epithelium has three parts that we talk now, and connective tissue. Then we will see what is epithelium. So epithelium is made up of stratified squamous epithelium. So what do you mean by the stratified? Stratified means that present in layers. So this epithelium is present in layers, and squamous means morphology of the cells. So it has different layers. The epithelium has different layers. The base layer is the stratum basale, and the upper layer is the stratum spinosum, then granulosum, then stratum corneum. So these cells undergo a process of maturation actually. So during which the cells produced by the mitotic division in the basal layer, they migrate to the surface where they are shed off and replaced by the maturing cell population. So these layers undergo a process of maturation, and this basal cells have a lot of mitotic activity because they produce new cells and then they go upwards. Then there are a lot of mechanism which are happening in between this maturation process that we will discuss. Sorry. So this is the basal layer. The cells made up of the basal layer cells are made up of keratinocytes. So they are the keratin producing cells. So once this keratinocyte enter the maturing compartment, they undergoes differentiation and becomes committed to certain biochemical and morphologic changes. So at the end of the differentiation process, a dead cell which filled with densely packed protein that contained within a toughened cell membrane is formed. So after reaching the surface, it is shed off. So that is known as the desquamation. So this process of migration of an epithelial cell from the basal cell layer to the surface is known as the maturation. So the time taken by a cell to divide and pass through the entire epithelium is known as the turnover time. And this turnover time is important because each area has different turnover rate. So if an area is having more turnover rate, has more immune power, it has more immune power, like that. So the cells at the basal layer are the least differentiated and contain the typical cellular organelles. And these cells, the cells in the basal layer, they synthesize DNA and they undergo mitosis and they produce new cells. And these cells, once they leave the basal layer, they become determined for the maturation. So that is how this process is happening. Then we will talk about the little more upper layer. So we told basal layer. What is this basal layer? And then next is the spinosum layer. So when we see about the morphology of the layer, this basal layer is little tall shaped, and when we go up, it become little more flattened and greatly flattened. So this is how the morphology goes. So we know I told you the function of the stratum basale. So where the production of cells happening. So then what is happening to the next layer? So the next layer is the stratum spinosum. So this is the basal layer, and stratum spinosum layer is this. So there what is known as the upper layer of stratum spinosum, they have certain Odland bodies. So that's the uppermost layer has Odland bodies. This is also known as keratinosomes or modified lysosomes. What happens is that they produce the acid known as the acid, there's an enzyme known as acid phosphatase. This acid they cause the destruction of the cell organelles. So they cause, inside the cell, we have the mitochondria, then what Golgi bodies, and all the other all parts are there inside the organelles. So what will happen? They destroy this. So upper layer has this acid phosphatase, but the concentration of acid phosphatase is maximum in the granulosome layer. So when the cell reaches this granulosome layer, they push the materials to the corner. So when we see that kind of cell, we can say that is known as the pycnotic nucleus, because they push the contents inside the cell towards the corner. So then that kind of cell is known as the pycnotic nucleus. So that can be seen in the stratum granulosum, because the concentration of acid phosphatase is more in the stratum granulosum. Then that is a feature of stratum granulosum, the pycnotic nucleus we told now. But the stratum spinosum has the Langerhans cells. What is Langerhans cells? If by mistake the bacteria crosses the corneum and granulosum and they reach the stratum spinosum, here is the Langerhans cells. The Langerhans cells are the neutrophil of the gingiva. It has antigen presenting cells. So this Langerhans cells has antigen presenting cells. What happens is that this Langerhans cells, they engulf the bacteria with the phagocytosis process, and the surface of APC, antigen presenting cell, we have the MHC, that is the major histocompatibility complex. So these MHC present on the surface of APC. So what is happening is that this MHC hold back the bacteria to show to immunoglobulins. So the immunoglobulins will come. So when the B and T lymphocytes come, they start producing antibodies against these bacterial antigens. So this is how the innate immunity works. So this process is happening in the stratum granulosum layer. And then one more thing is that these keratinocytes, they have precursors. So when they go to each layer, they have it is not this keratinocyte present not as it is not present as a directly as a keratinocyte. It has the precursor. So what is the precursor? So there are three precursors of keratin. They are the keratolynin, involucrin, and filaggrin. So this filaggrin has precursors that are packed into the keratohyalin granules. That's why when you go up, we can see the keratohyalin granules. So when we see this picture, you can see this is the keratohyalin granules. Inside that, the filaggrins are packed. They are the precursor. So when they go upside layer, we can see the keratohyalin granules and filaggrins. So above the basal layer, we told spinosum. There we can see the tonofilaments. They become denser here than the basal. So these can be seen in the spinosum layer, and above spinosum we told granulosum. So the cells of the granulosum are little more flatter and wider than the spinosum layer. So this contains certain basophilic keratohyalin granules. So and here the nuclei, I told the nuclei show certain signs of degeneration and pycnotic nuclei can be seen. And Ortolan bodies, these all are the features of the upper spinosum layer. Then above is the granulosum layer that I discussed now. Then uppermost layer is the stratum corneum. They are the larger layer and flatter than the granulosum layer. Then all nuclei and other organelles such as ribosomes, mitochondria, these all have disappeared in this stratum corneum layer. And keratohyalin granules are also disappeared. So the cells of the cornified layer are composed of densely packed filaments developed from the tonofilaments. They are altered and coated by the basic protein of keratohyalin granules, that is the filaggrin. So this keratinized cell becomes compact and dehydrated and covers a greater surface area than does the basal cells from which it developed, and finally this desquamates. So this is the process of overall stratified squamous epithelium. So certain things are happening. That is the proliferation and differentiation of keratinocytes. So these two things are happening that we discuss now. Overall I discuss, but I will talk in a concise manner. So this epithelium has a mechanical barrier. So how this barrier is happening is that they undergo proliferation and differentiation. So what is proliferation? The proliferation is a mitosis in the basal layer. So this mitosis less frequently can be seen in the suprabasal layers that I discussed now. Then next is the differentiation. So the process of keratinization has two parts, morphologic changes and biochemical changes. I told you the morphologic changes are the flattening of cells. I told you, no, this when we go up, the cells becomes little more flatter in morphology, and with the increased prevalence of tonofilaments. Then I told you the keratohyalin granules production becomes high when we go up, then the disappearance of nucleus. So these are the morphological changes. Then what is the biochemical changes? They are the increase in the TCA. TCA is the tricarboxylic acid. So this is highest in the basal layer because basal layer has more metabolism. So this TCA will produce in the basal layer is more. And then this pyruvic acid, this is the byproduct of the TCA cycle. So this pyruvic acid required for the keratin production. So these are the two changes, two things are happening in the gingival epithelium. So I think that you are clear with that. Then this keratinization. When we talk about keratinization, there are three types of keratinization are there. One is the orthokeratinization, then parakeratinization, then non-keratinization. So what is orthokeratinization? That is a complete keratinization because there is no nucleus in the stratum corneum. As we go upwards, the cells start destroy and filling with the keratin. So the lively cell, no, that may fill with the keratin. So keratin is the dead layer. So all organelles, they come on the side. So in stratum granulosum layer, this acid phosphatase destroy all the cell organelles. So this makes some space. So this nuclei has reached little more corner area. So that space is there. No, the remaining space, that space is filled with the keratin. So this is how the stratum corneum layer is full of keratin. And this orthokeratinization is maximum. This is a complete keratinization. So this keratinization is maximum in the palate, tongue and the gingiva, and least in the cheeks. And what is parakeratinization? Parakeratinization means there is no granulosum layer. So what all mechanisms are happening in the granulosum layer is not occur. So the stratum corneum retains the pycnotic nuclei. Actually, this pycnotic nuclei can be seen in the granulosum layer. But in parakeratinization, this granulosum layer is absent. So the stratum corneum, you can see this pycnotic nuclei, and also the keratohyalin granules are dispersed rather than giving rise to stratum granulose. So this is parakeratinization. Then what is non-keratinization? You know, so here is the complete absence of stratum granulosum and stratum corneum. So here corneum layer was there. Here the corneum and granulosum layer are absent. There is no keratin. So where and all we can see this non-keratinization? We can see in the sulcular epithelium and col and junctional epithelium. So here this is non-keratinized. In sulcular epithelium we can see the non-keratinization process, but it can be keratinized once it is exposed to the oral cavity or once the total bacterial flora of sulcus is removed, then it can be keratinized. So this is all about the keratinization. So next we will discuss about I think you all are clear with that, and when we will talk little more about the keratinocytes. So you know the keratinocytes are the principal cell types. They are present in at the basal layer, and these keratinocytes form keratin. So this keratin is a dead layer which provides immunity. So actually this dead layer, they cannot kill the organism, but they can push them. So the basal layer synthesizes keratin. The basal layer has low molecular weight keratin. When we go high, this low molecular weight keratin is converted to high molecular weight keratin, especially in the stratum granulosum layer. So that depend on the molecular weight, there are different keratin proteins. That is K1, 2, 10 and 12 that can be seen in ortho and para-keratinized area, and K16 and 6 can be seen in junctional epithelium, and 19 in para-keratinization area. Then K1 is the main component of the stratum corneum. So we discussed these all. Then I am going to talk about the cells of gingival epithelium. I told you, there are two types of cells. Keratinocytes we know now, we discussed, and there are some non-keratinocyte or clear cells are there in the gingival epithelium. So they are the melanocytes, Langerhans cells and the Merkel cells. And this actually what is the difference between this keratinocyte and non-keratinocyte is that in histologic sections these keratinocytes can be seen as acidophilic because they are visible as purple or dark color. But in non-keratinocytes, in histologic section, they lack keratin, so they appear lighter color. So this is known as clear cells. So we will discuss that. Actually this non-keratinocyte are not really important, so I just cover little bit of it. So what is melanocytes? This melanocytes present in the basal and the suprabasal layer. So this is the picture of melanocyte. They actually this keratinocytes has desmosomes. They are the connection between the two cells. But here the desmosomes and tonofilaments are absent, so they attach to the keratinocytes with the help of the dendritic process. Can you see this structure? This is the dendritic structure, and you can see this cell has this mitochondria, this nucleus, ref endoplasmic reticulum, Golgi apparatus. Golgi apparatus is a packaging apparatus. Then what is happening is that this is the melanin. So this ref endoplasmic reticulum, they synthesize melanin. This is the melanin. So in the Golgi bodies, what is happening is that this melanin pigment which is packed into vesicles. This is a vesicle. This is known as the melanosome. It has a plasma membrane outside, inside you can see the melanin. So this is the melanosomes. So these melanosomes, they move to the dendrites and then they reach the basal cells, and where they release this melanin granules, and they protect the nucleus or basal cells, and melanin pigment stored in melanocytes as melanosomes. We told So one melanocyte contact 30 to 40 keratinocytes. So one melanosomes, they create the melanosomes form in the Golgi apparatus. So this one melanosomes, they contact 30 to 40 keratinocytes. So since melanocytes are providing the cells of the keratinocyte with melanosomes, so the cell produced this cell provide mediators which are essential for normal melanocyte function. So usually these melanosomes, they present at the base of the interdental papilla. It can also be seen in the basement membrane, hard palate and all. So this is the melanocytes. These are one of the non-keratinocytes. Then next is the Langerhans cells. I just briefly told about Langerhans cells before. Actually, this Langerhans cells can be seen in the spinosum layer. This is a spinous layer. So inside you can see the Langerhans cells. It also has the dendritic process, and this is the basal layer. It has the antigen presenting cells I discussed just now, antigen presenting cells, MHC, and it has special bodies called Burbuck bodies. They push the content inside out. So this is the Langerhans cells. This also present in the basal layer. And next is the Merkel cell. So this Merkel cell responsible for tactile sensation, especially for light and touch. So as they lie close to the nerve terminal, they release certain neurotransmitters. So this is the keratinocyte and non-keratinocyte. Keratinocyte, when we go up, they decrease. The amount of keratinocyte decrease when we go upside layer. Then when we go up, what is happening? That flattening of cells, then tonofilament content will increase, then nucleus will disappear, then production of keratohyalin granules. This all happens when we go upside. When we go from basal layer to the corneal layer, these all things are happening. So this is the cells of gingival epithelium. So we covered that. So this is all about the gingival epithelium. So next remaining then we need to talk about junctional epithelium. So that I will explain in the coming video. So wait for that also. So if you have any doubt in this session, you can comment me in the comment section, then I will answer that. Thank you.